Today, I give you the gift of the amino acid alanine. It's the second smallest amino acid that has ever been seen. The smallest, well, that was glycine. As its side chain, this amino acid, this protein building block, only had a hydrogen, so basically just a space filler. With alanine, here we have a methyl group, so we have a carbon with three hydrogens. Okay, doesn't seem that exciting. And in fact, the kind of boringness of the side chain makes alanine useful for its genericness. If you look at these other amino acids, you see how they have like things like functional groups. They have maybe a hydroxyl group, or they have an amine group. They have a carboxylic acid. All these things that kind of allow them to have more interactions to maybe even do some covalent bond formation. Well, in the case of our methyl group, here alanine is really, really boring and it can't really do those things. And it's also pretty small. So it's second smallest if you compare it to all the amino acids. It's the second smallest after glycine. What this means is that if you want to see whether another amino acid in a protein is doing something, and you want to therefore like take away that side chain and see if that side chain was having an effect, well, you could take away the whole thing and just make it a glycine. You could do that with a technique called site-directed mutagenesis, where you basically go and you change the DNA sequence so that you change the amino acid in the protein and see does it have an effect. If you were to change it to a glycine, well, you might have an effect just because glycine has that structural loosey-goosiness. Because its side chain is so teeny tiny, its backbone is going to be more flexible than any of the other amino acids. With an alanine, well, here you're still going to get the benefit of being small, but you're not going to get the unbenefits of having that really loosey-goosey backbone. So it's going to behave more like the other amino acids in terms of the sorts of angles that its backbone will form. However, it's still going to be small and it's not going to have functional groups that will form unwanted interactions. And so by kind of doing an alanine scan, or by going through the amino acids and changing them one at a time to alanine, or maybe going and choosing a specific amino acid and changing it to alanine, that's going to allow you to see like what would happen if we took away the side chain of this amino acid. Was that side chain important? And by changing it to an alanine, well, now you're able to kind of see that. Often, however, alanine isn't actually the best choice. And so sometimes if you want to do something, you want to say, take a lysine and see if the charge is important. You might take that lysine and actually change it to something like a glutamine, or in the case of like a glutamate to a glutamine. These would be a better kind of size match if all you were caring about was whether that charge was mattering, whether that carboxylic group was mattering. But if you kind of just want to take away a side chain, well, mutating to an alanine is a good go for that. And that's thanks to alanine's kind of boringness. Going back to the idea of alanine being able to form those kind of more normal angles for a peptide backbone. So if you think about the peptide backbone, so we have all these amino acids lined up, and because of resonance in the peptide bond, there's only certain places where you can actually rotate in that backbone. With glycine, it can rotate a lot more than the other amino acids. With the big bulky amino acids, it can't rotate that well. With alanine, you're able to get a fairly flexible rotation, but not as flexible as glycine. And so you see it kind of adopt the normal angles. One of these kind of normal angles is for an alpha helix. And so that's one of these kind of common secondary structures where you have this hydrogen bonding between the backbones of the amino acids. And in this combination, that's going to give you these characteristic spirals, these alpha helices. Alanine loves these. It has the highest propensity for forming these alpha helices, and so you'll often find it in an alpha helix. Whereas you wouldn't find the glycine, and in fact, if you change something to a glycine that was in an alpha helix, you might disrupt the helix, which probably wasn't what you were going for if all you wanted to do was see if the side chain was doing something. Also, unlike glycine, alanine is chiral. And so it's unlike glycine in this respect, but it's similar to all the other amino acids in this respect. What I mean by chiral is basically it has right and left hand versions. And our bodies only use one of those versions. Our bodies use the L version. But because you have four different things attached to that central alpha carbon, you have kind of two different ways that that this can be sticking out. So you can think of like this methyl group sticking forward if things are oriented like this, or this methyl group sticking backwards if things are oriented like that. And those would be different forms. So you can see that if you have L-alanine versus D-alanine, you can't just flip over the D-alanine and look like the L-alanine. Instead, you'd have to actually break the bonds apart and swap them. And you can't just kind of flip it over. With glycine, you could just flip it over because there were two of the same thing attached to the central carbon. But with alanine, you can't. And so you can see this if you model things out. You can also kind of model things out if you think about putting rings on your fingers and looking and seeing the different ways you could rearrange your fingers without having to chop off your fingers and paste them back on. Chirality is really, really important. And so like it can make the difference between a molecule that's harmful and one that's helpful and one that's probably not helpful. Word of caution when it comes to the naming of these stereoisomers for these amino acids. In chemistry, when we're trying to like describe how the stereochemistry is laid out, we use this LD notation, where L stands for levorotatory, meaning if we shine polarized light at the molecule, that light is going to rotate to the left. And if we shine polarized light on a D molecule, the light would rotate to the right, dextrorotatory. This is not what it means in the case of amino acids. Instead, it's just saying that the orientation of that group sticking off of the alpha carbon is in the orientation of the L-glyceraldehyde. So originally this molecule glyceraldehyde, the smallest sugar, if you shine polarized light at it, at L-glyceraldehyde, it does turn left. And then they said, okay, well, let's just name all the amino acids following this convention. And so we'll say that all of these amino acids are going to be L because they're hooked up in that same way, even if they don't rotate light to the left. So alanine does not rotate light to the left. In fact, L-alanine is dextrorotatory, but L-serine is levorotatory. And so, yeah, that's not what the LD notation refers to. All the amino acids we use in our body are those L amino acids, even if they rotate light to the right. However, bacteria and bacterial cell walls, they actually do use D-alanine. So they use L-alanine and D-alanine in their peptidoglycan walls. And in fact, the beta-lactam antibiotics, so like penicillin, ampicillin, these take advantage of that. And they have the molecule that mimics the D-ala that's found in those peptidoglycan walls. And they get the enzyme that puts together those walls to attack the antibiotic. And then they get stuck on the antibiotic, and that causes the bacteria to have weak walls. So that was taking advantage of the fact that they have this D-ala structure, and we do not have that structure in our cells. You can see that if we take alanine and we were to kind of remove this amino group and stick a ketone in its place, so just like a carbonyl, what we would get would be this molecule pyruvate. The end product of glycolysis. And well, that pyruvate, you can decarboxylate it to give you acetyl-CoA. And you can take that acetyl-CoA and you can go through the tricarboxylic acid cycle, your citric acid cycle, and generate a lot of ATP. You could also take that pyruvate and go this way and make glucose. So we say that alanine is glucogenic, meaning we could be using it to make glucose. We take that pyruvate, we transform it to oxaloacetate, when we can use the oxaloacetate to make glucose. But in order to do that, we need to be able to remove this amino group and stick a carbonyl in its place. That would give us pyruvate. And then what are we going to do with this amino group? Thanks to transamination, we can remove that amino group and pass it on to a different molecule, a different alpha keto acid. So where you have a ketone in this alpha carbon. And typically we pass it on to alpha ketoglutarate, and that'll give us glutamate. And this way, we're able to safely transfer that amino group. And why is it important to safely transfer the amino group? Well, it's important because you don't want to just release that as ammonia, because then you get ammonia in your veins, you get Windex veins, not good. So instead, your body have this complicated process called the urea cycle, where they take those amino groups and they take them into the liver, pass it off to alpha ketoglutarate to give you glutamate, and you pass it off to oxaloacetate to give you aspartate. And then you take the amino groups from those and you pass them on together. And ultimately, you release them safely as urea. All of that is taking advantage of the fact that you can kind of transfer these amino groups from an amino acid to an alpha keto acid, and therefore finally transfer them on to make this molecule urea. Your bodies often send amino groups through your bloodstream on alanine and then transfer those amino groups onto alpha ketoglutarate in your liver where the urea cycle is happening. But those amino groups can be coming from any amino acid to start with. So you can take any amino acid, pass its amino group onto pyruvate, and that'll give you alanine. And then you can ship that alanine through the bloodstream and ship it to the liver, where then it can go and it can get passed off, the amino group can get passed off to alpha ketoglutarate to give you glutamate. So most of your tissues are going to actually send the amino groups through the body in the form of glutamine, but your muscles are going to send it in the form of alanine. And this is going to allow your muscles to do something called the glucose-alanine cycle. Your muscles, when they're working really hard, they're going to be breaking down proteins. And when they're breaking down proteins, they're going to be needing to remove those amino groups. Because, as we saw, the pyruvate can be used if they wanted to. It can be used in the citric acid cycle to make energy. And similarly, when you break down other amino acids, you'll get other alpha keto acids that then can enter the citric acid cycle and be used to make energy. In all of those cases, you've got those amino groups you got to get rid of. And so what the muscles do is they use the glucose-alanine cycle, where they pass the amino groups onto pyruvate. They use the skeletons to do whatever they want. Then they take that alanine through the blood, transfer the amino group off of the alanine onto the alpha ketoglutarate to make glutamate. And then the glutamate is going to go through the urea cycle and give you back urea. You're also going to be left with pyruvate when you take the alanine's amino group and put it back on alpha ketoglutarate. That gives you the pyruvate. And now what are you going to do with the pyruvate? Well, the muscles, they want the pyruvate back, but they don't just want pyruvate. They're like, hey, we want actual glucose. And the liver, it's able to do gluconeogenesis, so it's able to make glucose from pyruvate. And so it does that, and then it ships back glucose to the muscles, which is great because the muscles were breaking down proteins because they needed energy. And so now you're giving them some sugar, so they don't have to be breaking down their proteins. They can just break down the sugar. So voila, your glucose-alanine cycle is making your liver do the hard work for your muscles. And in the process, you're safely removing all the nitrogen. And this is taking advantage of alanine. And alanine transaminase, it's one of your liver enzymes. So this ALT, if you hear about doctors measuring like ALT and AST, AST is aspartate transaminase, ALT is alanine transaminase. Both of those are playing important roles in the urea cycle in your liver. And well, if your liver is damaged, then those enzymes can leak out into the blood. And then the doctors can measure that and be like, hey, those don't belong here. And then be like, your liver needs some help. So the glucose-alanine cycle, awesome. More on that elsewhere. Now let's talk a little about the history of alanine, or at least the discovery of alanine. So alanine's been around for a really, really, really long time. But the first time it was kind of made from scratch, other than in a body, was in 1850. It was one of two protein amino acids that was made from scratch this way before it was actually shown to be a proteinogenic amino acid, like that it was a part of a protein. This guy, Adolf Strecker, he made it and he named it alanine, with the German using the first syllable of the word aldehyde to denote its origin. So he made it from aldehyde ammonia, and he was trying to make lactic acid, but he accidentally made alanine, and so thankfully he did. And so as to aldehyde ammonia, so remember an aldehyde is just a carbonyl with a hydrogen on the side. And so he was making it from aldehyde ammonia, and he made this alanine. Voila. The accidents that give us such cool stuff. As for actually isolating it from a protein and proving that it was from a protein, that's complicated. So remember that the synthesis was happening in 1850. In 1875, Schutzenberger and Bourgeois isolated it from base hydrolysis of silk, or they say so. They didn't rigorously analyze it, but it seemed to be that alanine. And a few years later, Schutzenberger did some more thorough analysis, And when they first did that base hydrolysis, they found there was a bunch of stuff in there, and he further separated the bunch of stuff by fractional crystallization. So using different chemicals and to isolate things by getting them to crystallize or solidify under different conditions and separate the different components. And then he measured how much carbon, nitrogen, and hydrogen each of the fractions had. One of them was consistent with alanine, but again, he didn't do any more tests on it. So we didn't have proof that those atoms were actually hooked up in the alanine way. Then in 1888, Theodore Wheel purified it from hydrolysis of silk. And so silk is really, really rich in alanine, so it was great for this. So that's why they're kind of like both were using it. And then he characterized it, and he characterized it more, and so he came credit. I'm not one to care about credit. Science is a team effort, and every contribution helps. What I care about is what is found out, so that's what I want to tell you about. Or I guess I did tell you about, now this is at the end. Yeah, I wrote this a while ago. But that's alanine for you. So a couple of the key things, it's going to be the second smallest. It is chiral, unlike glycine. It's boring. It's generic. It is most likely to form an alpha helix. It's great if you want to do some site-directed mutagenesis and see if an amino acid is important. Remove that amino acid and just have an alanine there, and voila. Well, I mean, alanine is still an amino acid, but if you want to see if the side chain is important, alanine has the smallest side chain other than glycine, and glycine is all loosey-goosey, whereas alanine is kind of more normal, more generic, more blah. But it does have the bragging rights of having the highest propensity to form an alpha helix. And it's also super duper important for transporting amino groups through the bloodstream, especially from your muscles to your liver, so that your muscles can get rid of nitrogen groups when they're breaking down protein and get back glucose in the glucose-alanine cycle. So there you have it, all about alanine.